The era when Earth's oxygen nearly killed everything. Beneath a sky thickening with invisible danger, Earth's first breaths of oxygen spread silently across oceans and continents, reshaping every molecule of life. Billions of years ago, a once benign atmosphere turned deadly, triggering a slow-motion apocalypse that spared few and transformed all. Microbial mats flared with new chemistry, while fragile anaerobes clung to shrinking refuges, their survival hanging on molecular ingenuity. This was a world caught between destruction and possibility, where energy and poison were the same. Join us as we explore the era when the very air became both enemy and architect of life's future. When the sky first turned hostile, a planet long wrapped in a chemically gentle sky began to change when free oxygen accumulated beyond trace amounts, crossing a threshold that had no visible boundary, yet carried irreversible consequences for every surface it touched. Geological proxies indicate that between roughly 2.4 and 2.1 billion years ago, atmospheric oxygen rose from less than 1 part per million to levels high enough to disrupt global chemistry, a shift detected today through the disappearance of mass-independent sulfur isotope fractionation in ancient sediments. This subtle isotopic silence marks the moment when oxygen stopped being locally produced and immediately consumed, and instead lingered long enough to alter the balance of the air itself. For countless generations of early life, the sky had been chemically neutral, composed largely of nitrogen, carbon dioxide, and methane, gases that neither corroded proteins nor stripped electrons from fragile molecular bonds. Oxygen, however, behaved differently the moment it accumulated, because its strong electronegativity allowed it to react aggressively with reduced compounds that had dominated Earth's surface for more than 1 billion years. Even at concentrations far below modern levels, oxygen destabilized the equilibrium that anaerobic metabolisms relied upon, transforming the atmosphere from a passive medium into an active environmental force that imposed new energetic costs on every exposed organism. High above the surface, sunlight became an accomplice rather than a neutral witness, as oxygen altered atmospheric photochemistry in ways that early life could not escape. The presence of free O2 allowed ultraviolet radiation to generate highly reactive byproducts, including atomic oxygen and early forms of ozone, which changed how radiation penetrated the lower atmosphere. While modern ozone shields life from lethal ultraviolet wavelengths, early and unstable ozone formation likely created fluctuating radiation regimes, exposing surface ecosystems to intermittent UV stress rather than consistent protection. Experimental reconstructions suggest that these conditions intensified DNA damage in exposed microbial communities, linking oxygen accumulation not only to chemical toxicity, but also to increased mutational pressure, driven by altered atmospheric light filtering. Chemical modeling suggests that once oxygen levels reached even 0.001% of the present atmospheric concentration, spontaneous reactions with methane accelerated, reshaping atmospheric composition within relatively short geological intervals. Methane, which had provided powerful greenhouse warming under a faint young sun, began to oxidize into carbon dioxide and water vapor, thinning the planet's thermal blanket and altering photochemical reactions high above the surface. These reactions did not occur uniformly because ultraviolet radiation, atmospheric circulation, and localized biological production created oxygen oases that expanded and collapsed over time, producing a sky whose hostility arrived unevenly but relentlessly. The biological consequences of this shift were immediate and profound because oxygen penetrated cellular membranes and generated reactive oxygen species capable of damaging DNA, proteins, and lipid structures. Most microorganisms alive during this interval lacked enzymatic defenses such as superoxide dismutase or catalase, leaving them vulnerable to oxidative stress even at concentrations that would seem trivial by modern standards. Laboratory studies of extant obligate anaerobes demonstrate how oxygen disrupts iron-sulfur clusters and enzyme-active sites, providing a mechanistic window into why early life experienced oxygen not as nourishment, but as an invisible toxin diffusing through the air. Geological records reinforce this biological narrative by revealing widespread oxidative alteration of surface minerals that had previously remained stable under reducing conditions. Paleosols, preserved from this interval, show the first appearance of ferric iron oxides formed directly through atmospheric exposure, indicating that oxygen was no longer confined to localized microbial mats or shallow waters. These oxidized soils represent a chemical dialogue between air and land, where oxygen stripped electrons from minerals long shielded from such reactions, signaling that the sky itself had become an agent of transformation rather than a neutral canopy. Atmospheric circulation further amplified oxygen's reach because winds transported reactive gases across continents and oceans, preventing isolated refuges from remaining chemically untouched. Even regions distant from biological oxygen production felt its effects as photochemical reactions generated oxidants capable of traveling long distances before decaying. This meant that survival increasingly depended not only on location but on molecular resilience, forcing life to confront a planet where the very act of exposure to air carried escalating risks that could no longer be avoided through simple spatial retreat. Climate feedbacks compounded these pressures as declining methane levels weakened greenhouse warming, allowing global temperatures to drop despite the continued luminosity increase of the Sunday. Sedimentary evidence points toward expanding glacial deposits during this same interval, suggesting that atmospheric oxygen indirectly contributed to climatic instability while life struggled to adapt to its chemical presence. The sky, once a stabilizing buffer, now exerted control over temperature, radiation filtering, and chemical weathering, binding biological survival to atmospheric processes that operated far beyond the scale of individual organisms. Isotopic fingerprints, preserved in ancient carbonates, reveal that oxygen's growing presence also disrupted global carbon cycling while the atmosphere was still chemically unstable. Shifts in carbon isotope ratios suggest changes in biological productivity and burial rates, indicating that oxygen altered not only what gases filled the sky, but also how efficiently carbon moved between air, ocean, and rock. These disruptions fed back into atmospheric composition by modulating carbon dioxide levels, subtly influencing climate and ocean chemistry at the same time that oxygen toxicity strained existing life. The sky's transformation, therefore, extended beyond direct biological harm, reshaping the planet's fundamental exchange systems, while equilibrium remained elusive. Yet oxygen did not arrive as a sudden flood, but as a rising tide whose persistence mattered more than its speed, because the damage it inflicted accumulated over generations rather than moments. Each incremental increase widened the gap between organisms capable of tolerating oxidative stress and those trapped by metabolic pathways that had evolved under permanently oxygen-free conditions. This gradual escalation transformed natural selection into a chemical filter where survival increasingly depended on the ability to endure an atmosphere that no longer merely surrounded life but actively reshaped its limits. By the time oxygen established itself as a lasting component of the sky, Earth had entered an era where the atmosphere imposed consequences before land or climate could respond fully, turning air into the first frontier of planetary upheaval. The transformation did not announce itself with storms or flames, but with silent reactions unfolding molecule by molecule above the surface, as gases rearranged and sunlight catalyzed unfamiliar pathways. As this altered air settled into a new equilibrium, its excess did not linger harmlessly overhead because gravity and circulation carried its chemical force downward, where the vast and iron-rich oceans waited to absorb the shock and reveal how deeply the planet would be changed. Oceans that began to rust. Gravity ensured that the consequences of a changing sky did not remain aloft because oxygen dissolved readily into the planet's shallow seas the moment it accumulated above them. As atmospheric oxygen reached persistent concentrations, surface waters absorbed it according to temperature gradients, salinity, and partial pressure, allowing dissolved O2 to spread laterally across continental shelves and microbial mats. This transfer required no storms or dramatic mixing, because simple diffusion was enough to introduce a chemically aggressive element into oceans that had remained reducing for more than 1 billion years. Once dissolved, oxygen did not linger passively in seawater because the marine environment was primed for reaction, ensuring that its arrival would trigger immediate and far-reaching chemical consequences across multiple oceanic basins. Long before waves broke against continents recognizable today, seawater carried immense quantities of dissolved ferrous iron, released continuously from volcanic crust and hydrothermal systems. Mid-ocean ridges, submarine volcanoes, and fissures in the early seafloor supplied vast amounts of phi-squared mice into the water column, while the near absence of oxygen allowed this iron to remain soluble across enormous distances. Iron-rich oceans were not marginal or isolated environments, but the dominant marine condition, supporting microbial communities that relied on iron for energy transfer, enzymatic function, and structural stability. This abundance shaped early metabolism so deeply that iron became embedded in life's most fundamental biochemical pathways long before oxygen appeared as a competing force. When oxygen encountered this iron-rich chemistry, electrons shifted abruptly, converting soluble ferrous iron into insoluble ferric minerals that could no longer remain suspended in seawater. The oxidation of Fe² mice into Fe³ mice produced iron oxides and hydroxides that aggregated into solid particles almost instantly under marine conditions. These reactions occurred spontaneously and at scale, stripping dissolved iron from seawater far faster than biological systems could compensate. What had once circulated freely through the oceans as a nutrient and metabolic tool was abruptly transformed into a chemical liability, precipitating out of solution as oxygen advanced into previously stable marine zones. These reactions unfolded across entire ocean basins, producing vast clouds of iron-rich particles that drifted downward through the water column like slow, rust-colored snowfall. The process was uneven, shaped by fluctuations in oxygen production, water depth, and proximity to photosynthetic communities, yet its cumulative effect was relentless. Iron oxides accumulated on the seafloor in quantities large enough to alter marine chemistry for millions of years, permanently removing bioavailable iron from circulation. The oceans, once transparent and chemically generous, began to lose one of their most essential dissolved elements through a process that left no immediate biological recovery. Marine organisms were therefore affected even when they never directly encountered oxygen, because the chemistry of the water around them was being stripped of the elements their metabolisms required to function. Layer by layer, the seafloor recorded this transformation in banded iron formations, whose alternating mineral bands preserve a rhythmic history of oxygen pulses and chemical collapse. These formations, composed primarily of hematite, magnetite, and silica-rich chert, reflect repeated oscillations between oxidizing and reducing conditions in the ancient oceans. Radiometric dating places many of these deposits between roughly 2.7 and 1.9 billion years ago, tightly bracketing the era of rising atmospheric oxygen. Each band represents not merely sediment, but a moment when chemistry tipped decisively, locking iron into stone and withdrawing it from biological reach on a planetary scale. Less visibly, iron oxidation altered seawater chemistry itself, shifting pH balances and trace metal availability in ways that compounded biological stress. As iron precipitated, it scavenged other dissolved elements, including phosphate and trace metals essential for cellular function, further reducing nutrient availability in surface waters. These cascading chemical effects meant that oxygen did not simply remove iron, but reorganized marine chemistry into a less hospitable system overall. The oceans became chemically selective environments where multiple limitations overlapped, tightening the constraints under which early life was forced to operate. For microorganisms adapted to iron-rich conditions, the loss of bioavailable iron disrupted enzymes central to respiration, nitrogen fixation, and carbon metabolism. Iron-sulfur clusters within proteins became increasingly difficult to assemble, weakening electron transport chains and slowing cellular energy production. Anaerobic organisms that relied on iron as an electron donor or acceptor found themselves constrained by scarcity rather than toxicity alone. Survival now required operating metabolic systems with fewer functional components, forcing life to function under biochemical limitations it had never previously encountered. Even oxygen-producing cyanobacteria faced an emerging paradox, because the machinery of oxygenic photosynthesis depended heavily on iron-rich proteins. Photosystem 1 and 2 contain iron cofactors essential for electron transfer, making iron limitation a direct constraint on oxygen production itself. As iron availability declined in surface waters, cyanobacterial growth rates fluctuated, introducing feedback loops that destabilized primary productivity. The organisms reshaping the planet were simultaneously undermined by the consequences of their own success, preventing rapid stabilization of marine ecosystems. Ocean stratification intensified these effects because oxygenated surface layers became increasingly isolated from deeper, iron-rich anoxic waters. Density gradients limited vertical mixing, trapping oxygen near the surface while preserving reducing conditions below. Although deeper waters retained dissolved iron, they lacked sufficient light to support photosynthesis, cutting off access to energy sources required for complex metabolic activity. The oceans fractured into chemically distinct layers, each governed by incompatible conditions that constrained adaptation and narrowed ecological possibilities. Over millions of years, this vertical separation transformed marine ecosystems into chemically partitioned worlds, where survival depended less on geography than on elemental access. Shallow waters became oxidized and iron-poor, favoring organisms capable of tolerating scarcity and oxidative stress, while deeper zones served as refuges for anaerobic life operating under severe energetic constraints. Evolutionary pathways narrowed as entire metabolic strategies became untenable, not through sudden extinction, but through prolonged chemical exclusion that reshaped life's options. As iron vanished from circulation and settled permanently into stone, life confronted a quieter catastrophe, one defined not by poison alone, but by the disappearance of a foundational resource. The oceans absorbed oxygen's chemical force by locking away an element woven into the fabric of early biology, transforming abundance into absence without spectacle or warning. What remained was a biosphere forced to adapt to scarcity imposed by planetary chemistry itself, where survival no longer depended solely on avoiding harm, but on enduring a world that had quietly taken away one of life's most critical tools, a poison to the first breathers. Oxygen, once a silent chemical force in the seas, began infiltrating the interior of early cells, where its presence had never been anticipated. For organisms that had evolved entirely under reducing conditions, even trace amounts of molecular oxygen were disruptive, penetrating membranes and encountering biochemical systems unprepared for its reactivity. The arrival of oxygen was invisible but inexorable, and within each cytoplasm, atoms of a gas that had reshaped planetary chemistry now threatened the very machinery that enabled life to function. Across oceans and shallow pools alike, cells confronted a new world, one where an essential element in excess had become a molecular adversary capable of destabilizing the delicate balance of early life. Molecular oxygen reacted with cellular components to generate reactive oxygen species that attacked membranes, proteins, and nucleic acids with relentless intensity. Superoxide anions, hydrogen peroxide, and hydroxyl radicals emerged spontaneously in metabolic reactions and light-driven processes, tearing apart molecular bonds that had never before been challenged. Membranes, composed of lipid bilayers and embedded proteins, were particularly vulnerable as peroxidation altered fluidity and permeability, causing leakage of ions and metabolites. Proteins containing iron, sulfur, and other redox-active cofactors were especially susceptible, collapsing their three-dimensional structure and impairing enzymatic function, leaving cells scrambling to maintain internal order against an invisible chemical storm. Critical iron-sulfur clusters, central to early enzymatic reactions and energy transfer, became vulnerable targets for oxidative cleavage. These cofactors, embedded within ferredoxins and other metalloproteins, facilitated electron transfer in respiration and nitrogen fixation. When oxygen infiltrated the cytoplasm, electrons were stolen from these clusters, destabilizing protein function and halting essential biochemical reactions. Metabolic pathways that had operated reliably for millions of years faltered, producing energy deficits and interrupted nutrient cycling. Life's most foundational chemical processes, once assumed permanent, now faced relentless disruption, illustrating how oxygen's threat was not merely external, but molecular and inescapable. DNA molecules accumulated damage from radicals and free oxygen, destabilizing the genome and increasing mutation rates far beyond natural replication errors. Single and double-strand breaks, base modifications, and cross-linking emerged as radical attacks outpaced cellular repair. The genomes of early prokaryotes were compact but highly efficient, and even minor lesions had disproportionate consequences on replication and transcription. Mutations occurred not in isolation but in waves across populations, introducing instability while simultaneously creating the potential for adaptive novelty. Cells faced a paradox. Oxygen was both destructive and, in small doses over time, a driver of evolutionary change through increased genomic variability. Without protective antioxidative mechanisms, the internal chemistry of cells became a battlefield where structural and metabolic systems were constantly challenged. Enzymes such as catalase, superoxide dismutase, and peroxidases, which in later life forms mitigate oxidative damage, had not yet evolved. Consequently, any oxygen that entered a cell became a reactive intruder, capable of damaging multiple targets simultaneously. Even highly robust proteins and RNA molecules suffered, leading to cascading failures across metabolic networks. This chemical pressure forced cells into reactive stasis, a state where survival depended on tolerating molecular assault rather than optimizing growth or reproduction. Even simple electron transport chains and redox-active cofactors were disrupted, halting energy production and nutrient assimilation across microbial populations. Cytochromes, quinones, and ferredoxins lost efficiency under oxidative stress, compromising ATP generation and preventing the proper cycling of electrons necessary for respiration and photosynthesis. Nutrient assimilation slowed, and essential metabolites became scarce, producing bottlenecks in growth and replication. Populations that had once thrived under stable, reducing conditions now faced an energy-limited existence, where the cellular machinery that had sustained life was slowly being undermined from within. Microbial communities adapted to stable, reducing conditions now faced patchwork survival, with some lineages shrinking while others sought chemical or spatial refuges. Anaerobic organisms retreated to deeper or isolated niches where oxygen had not yet penetrated, while surface-dwelling cyanobacteria and chemoautotrophs confronted the chemical challenge directly. Competition intensified as habitats became more heterogeneous, favoring species capable of tolerating partial oxygen exposure or exploiting oxygen-free microenvironments. The uneven pressures created early forms of ecological segregation, fragmenting populations into pockets of survival and decline that foreshadowed later evolutionary diversification. Gradients of oxygen across shallow and deeper waters fragmented populations further, creating zones of intense oxidative stress and restricted habitat. Oxygen-rich surfaces favored species capable of rudimentary detoxification or rapid repair, while intermediate zones became hostile to most life forms. Deep anoxic layers remained refuges, but isolation prevented gene flow, slowed evolutionary innovation, and limited resource availability. The oceans became mosaics of habitability, where survival no longer depended solely on nutrient acquisition, but on the capacity to endure a toxic chemical environment that threatened molecular integrity at every turn. Physiological strain reshaped interactions between species, altering competition, symbiosis, and reproductive success, quietly remolding the architecture of early ecosystems. Organisms that could tolerate oxygen began exploiting niches left vacant by less adaptable species, while symbiotic relationships adjusted under new stress regimes. Population densities fluctuated unpredictably, altering predation and nutrient cycling. Even microbial mats and biofilms, previously considered stable communities, became fragile and patchy under oxidative assault. Ecosystems began fracturing externally, reflecting the molecular pressures each cell experienced internally. While individual cells struggled to maintain molecular integrity, evolutionary pressures began to favor novel defenses, hinting at adaptations that would allow life to endure an oxygen-rich world. Some lineages developed rudimentary mechanisms to detoxify reactive oxygen species or compartmentalize sensitive biochemistry. Others relied on behavioral strategies, such as moving to low-oxygen niches or forming protective colonies. Life endured not because oxygen was benign, but because selective pressures demanded ingenuity and resilience. Yet the fragility of ecosystems mirrored the vulnerability of each cell, showing that while organisms survived internally, entire communities were now on the brink of structural and ecological transformation, leaving the stage set for a world increasingly shaped by chemical toxicity. The collapse of the methane world. The atmosphere itself became a battlefield as a once stable veil of greenhouse gases faced chemical upheaval from a newly pervasive element. What had long kept the planet warm, vast clouds of methane suspended above oceans and continents, now reacted relentlessly, producing water and carbon dioxide in quantities the Earth had never experienced. The balance that had nurtured microbial life for billions of years shifted invisibly but inexorably, and the climate began to feel the pressure of chemical change that no organism could control. Across the globe, the faint, silent reactions of gases in the sky foreshadowed an environmental transformation as sweeping as any cellular upheaval. Methane molecules, potent and abundant, collided with oxygen in a series of chemical reactions that converted them into carbon dioxide and water. The oxidation proceeded with a half-life of only a few thousand years, fast enough on geological timescales to thin the greenhouse layer dramatically. Trace products, including hydroxyl radicals, briefly intensified atmospheric reactivity, creating transient chemical zones that rippled across latitudes. Methane concentrations, estimated at thousands of parts per million in the late Archean, fell sharply, altering the balance of radiation absorbed and emitted by the planet. These molecular exchanges, invisible to the eye, had consequences that were planetary in scale, transforming the warmth of oceans, the circulation of air, and the very conditions under which life had evolved. With the loss of methane, the greenhouse effect that had long stabilized surface temperatures began to unravel. Global mean temperatures may have dropped by as much as 10–15 degrees Celsius, a dramatic shift in a world previously warmed by potent methane concentrations. Even minor decreases in trapped infrared radiation destabilized shallow seas and surface pools, altering evaporation rates and nutrient cycling. Organisms accustomed to stable warmth faced a cooling environment where metabolic processes slowed and growth rates declined, forcing early life to confront a climate now dictated by chemistry rather than habit. The thermal retreat of habitable zones created patchy refuges, fragmenting communities across continental shelves and shallow oceans. Geological records bear witness to this upheaval, preserving the chemical fingerprints of a shifting atmosphere. Carbon isotopic ratios in ancient sediments reveal marked excursions in delta to the power of 13 C values, signaling the rapid removal of methane and the redistribution of carbon in oceans and rocks. Banded iron formations, previously precipitated during oxygenation events, now capture indirect evidence of atmospheric change as iron cycling interacted with newly available oxygen and CO2. These isotopic and mineralogical markers serve as a silent archive of the planet's transformation, revealing the scale and pace of a crisis written in stone and seawater. They also hint at the intricate feedback between atmospheric chemistry and early oceanic life, connecting the molecular with the macroscopic. Microbial communities that once thrived under anoxic conditions faced indirect but relentless pressures. Methanogenic archaea, which had flourished by producing methane in sediments, suddenly confronted a world where their metabolic output was destroyed before it could accumulate. Methanotrophs, capable of consuming methane, experienced disrupted niches and chemical stress from oxygen and CO2 fluctuations. Methane-dependent microbial food webs collapsed in areas where gas flux declined most sharply, forcing microbial mats and biofilms to reorganize around surviving methane pockets. These ecological reorganizations reshaped energy flow, reproduction, and competition, illustrating how atmospheric chemistry dictated survival long before visual signs of stress appeared. Atmospheric circulation propagated these chemical changes unevenly, generating gradients of oxygen and methane across the globe. High-latitude regions, receiving less solar energy, experienced accelerated cooling, while equatorial zones retained slightly more warmth but still faced diminishing greenhouse buffering. The interplay of chemistry and circulation produced complex climate mosaics, where localized conditions diverged sharply from global averages. Life in shallow seas, coastal pools, and sedimented basins experienced a spectrum of stress intensities, forcing dispersal, migration, or local adaptation in an environment no longer uniform or forgiving. Microbial and algal populations in stratified waters faced nutrient shortages as circulation slowed and oxygen penetrated previously anoxic zones. Ice and snow expanded in response to the thinning greenhouse blanket, triggering positive feedback loops that amplified cooling. Increasing albedo reflected more sunlight into space, reinforcing temperature decline and extending glacial coverage. Dropstones, embedded in ancient bedrock and striated continental surfaces, hint at early large-scale glaciations, silent testimonies to atmospheric transformation. As sea ice advanced, it limited surface mixing, reducing nutrient exchange with deeper waters and adding chemical stress to already struggling microbial populations. Surface oceans cooled, evaporation slowed, and chemical stratification intensified, forcing early life to occupy increasingly constrained habitable zones. The interplay between atmospheric chemistry and planetary temperature reshaped weather patterns and ocean currents. With methane levels collapsing, winds and precipitation regimes became more variable, destabilizing nutrient flows and redistributing thermal energy unevenly across latitudes. Ocean stratification intensified, separating oxygen-rich surface waters from colder, deeper layers where methane lingered temporarily. The combination of chemical, thermal, and circulation shifts created novel selective pressures, sculpting both microbial populations and early ecosystems into forms capable of surviving under new climatic constraints. Some coastal and shallow water communities found temporary relief, while others vanished entirely, leaving empty niches to be filled in subsequent eons. Life's vulnerability was made clear by the cascading consequences of a single chemical change. Organisms that had survived the molecular threats of oxygen now faced the indirect but equally potent force of climate instability. Reproduction slowed, habitats contracted, and interspecies interactions were altered as communities fragmented. The collapse of methane illuminated the interconnectedness of planetary chemistry, climate, and biology, showing that even a gas invisible to the eye could dictate the fortunes of life on a global scale. Fragile ecosystems revealed that molecular and atmospheric shifts were inseparable, and survival required adaptation across both microscopic and planetary dimensions. As methane vanished and the planet cooled, the climate responded swiftly and not gently, establishing conditions that would define the following era of Earth's history. Ice sheets advanced, surface waters chilled, and ecosystems were forced to reorganize under selective pressures unlike any they had previously experienced. The stage was set for a biosphere in flux, where survival demanded ingenuity, adaptability, and resilience at scales both microscopic and planetary. The world had become chemically, thermally, and ecologically altered, leaving early life to navigate a climate dictated by the very reactions that had once silently reshaped the seas. Ice advancing under an oxygen Sunday, a sudden stillness fell over the planet as temperatures plunged and ice began to claim previously temperate regions. The retreat of methane's insulating embrace, combined with the unyielding presence of oxygen, left Earth's surface vulnerable to solar energy loss, initiating an era of planetary-scale freezing. Crystalline layers reflected faint sunlight, casting shimmering patterns across shallow lagoons and tidal flats. Coastal pools, once teeming with microbial mats, froze into silent tableau, where life persisted only in the slimmest margins. Glaciers crept outward from high latitudes, gradually thickening and extending toward equatorial zones, reshaping landscapes that had once been warm, shallow, and chemically reactive. These early ice sheets were not mere blankets of frost, but active geological forces, scouring rock, redirecting meltwater, and establishing physical barriers that constrained life in ways chemical toxicity alone could not achieve. Sheets of crystalline frost spread across continents, slowly thickening into glaciers that reshaped coastlines and river valleys. As ice advanced, it ground underlying bedrock into fine sediment, transporting debris toward the oceans and leaving striations and grooves as enduring records of motion. Glacial grinding not only sculpted valleys but pulverized bedrock into silt and sand that oceans later carried, subtly enriching nutrient concentrations in localized basins. The sheer weight and movement of these glaciers depressed continental crusts, altered drainage patterns, and temporarily redirected river systems, forcing microbial and algal populations in coastal and inland waters to navigate fragmented, ephemeral habitats. Surface waters cooled dramatically, limiting both photosynthetic activity and nutrient circulation, while frozen landscapes became stark arenas of environmental constraint. Striated bedrock and dropstones embedded in ancient sediments offer a frozen record of this early glaciation, marking the reach and movement of ice across land masses. Diamictites and tillites preserve the evidence of sediment transported by glaciers, while embedded boulders testify to the destructive energy of ice-driven transport. These physical markers, coupled with chemical signatures in surrounding rocks, indicate that glaciers were not stationary but dynamic, advancing and retreating over millennia in response to subtle shifts in temperature and atmospheric composition. They reveal a planet in flux, where chemical, thermal, and physical forces interact in a delicate and often violent equilibrium. Meltwater channels cut through glacial beds, leaving behind sedimentary hints of ephemeral rivers and pools where life could temporarily persist. Rising reflectivity, or albedo, from expanding ice-reinforced cooling, creating feedback loops that accelerated the advance of frost. Sunlight reflected by white, crystalline surfaces reduced solar heating, extending ice coverage further and faster than chemistry alone would dictate. In shallow seas, this effect slowed evaporation, concentrated salts, and further altered chemical gradients, challenging microbial communities already stressed by oxygenation and methane depletion. The planet's surface became a mirror, reflecting both light and the fragility of ecosystems, as physical and chemical pressures compounded to create an inhospitable, frozen world. Thin ice in marginal zones created delicate, transient habitats, where only the most resilient microbial mats could exploit fleeting warmth and light. Shallow seas and coastal lagoons experienced chilling and stratification, isolating microbial communities and altering nutrient cycles. Cyanobacterial mats, once flourishing in oxygen-poor shallow waters, were trapped beneath a thin layer of ice or confined to marginal pools warmed by geothermal activity. Stratification slowed vertical mixing, limiting the availability of dissolved gases and minerals while concentrating metabolic byproducts in confined spaces. Some cyanobacteria slowed photosynthesis, forming dormant layers, while chemoautotrophs exploited mineral-rich sediments beneath ice shelves. Microbial respiration became spatially patchy, creating pockets of activity amid broad inactivity. These environmental pressures created evolutionary bottlenecks, favoring microbes capable of tolerating low temperatures, low light, and highly variable chemical conditions, and forcing others to perish or retreat into refugia. Oceans, once teeming with chemically stressed microbes, now faced physical barriers of ice, slowing circulation and trapping gases below the surface. Methane that survived in isolated pockets was further oxidized in an increasingly oxygen-rich environment, creating localized zones of acidity and chemical stress. Marine life adapted to stable temperatures now contended with both chilling and oxygen-driven oxidative reactions, testing the limits of cellular resilience. The coupling of physical barriers with ongoing chemical pressure demonstrates that freezing was not merely an inert state, but an active challenge to life, demanding behavioral, structural, and metabolic ingenuity. Some microbial communities adapted by forming dense biofilms beneath ice shelves, while others survived only in transient meltwater pools. Regional variation in glacial extent created a mosaic of habitable refuges and frozen deserts, pushing life into pockets of survival and adaptation. Equatorial and geothermal refuges provided transient warmth and chemical stability, allowing some microbial mats and early eukaryotes to persist, while mid-latitude oceans and continental interiors became largely uninhabitable. This patchwork of habitable zones promoted genetic diversification and localized adaptation, a prelude to the evolutionary innovations that would later exploit oxygenated and chemically complex environments. Isolation and environmental stress became engines of both survival and transformation. Seasonal or localized melt zones offered fleeting opportunities for reproduction, temporarily sustaining fragile communities amid vast ice-bound expanses. Evidence of repeated glacial advances suggests that early Earth oscillated between cold extremes and partial thaws, hinting at the planet's fragile thermal balance. Sediment cores reveal cycles of deposition indicative of transient melting and ice retreat, suggesting that climate instability was punctuated rather than uniform. These fluctuations created temporal windows for microbial and early eukaryotic expansion, allowing life to exploit favorable conditions before cold extremes returned. Periods of partial thaw allowed fleeting blooms of metabolic activity, only to be curtailed by renewed ice expansion, illustrating the delicate balance between chemical forcing and physical restraint. The oscillating nature of early glaciation underscores how oxygen-driven chemical changes were intertwined with physical forces to generate a dynamic, challenging biosphere. Chemical transformations continued beneath the ice, as oxygen and trace metals interacted with sediments, sustaining subtle but ongoing metabolic and geochemical cycles. Iron and sulfur compounds underwent slow oxidation, while microbial mats metabolized limited nutrients trapped beneath frozen surfaces. Even under kilometers of ice, life persisted in niches shaped by chemistry rather than light, demonstrating resilience and adaptation in the face of extreme environmental constraints. These hidden cycles foreshadowed the future interplay between oxygen, nutrients, and biological innovation that would eventually reshape Earth's biosphere. Microbial respiration and chemical reactions persisted in isolation, producing microenvironments that would serve as cradles for future evolutionary experimentation. Cold-locked ecosystems remained in place, yet the planet's chemistry quietly pressed onward, hinting at pressures that would soon drive the next evolutionary chapter. Frozen seas and expansive ice sheets immobilized habitats, but oxygen continued to circulate. Iron and trace metals persisted in reactive forms, and subtle chemical reactions persisted beneath the surface. Life endured, constrained and cautious, as the environment imposed new limitations and prepared the stage for transformations that would follow. The world had become both still and dynamic, frozen in appearance yet chemically restless, leaving organisms to navigate the delicate tension between survival and the quiet, relentless march of planetary change. Extinction without fossils. Silent mortality swept through ecosystems, leaving no visible trace of its devastation across frozen seas and oxygenated waters. The advancing oxygen-rich environment continued to alter chemical and thermal landscapes, pushing fragile, microbial and soft-bodied communities past the limits of survival. Where glaciers immobilized surface habitats, reactive oxygen species penetrated the oceans and shallow lagoons, disrupting delicate metabolic machinery that had evolved under low oxygen conditions. The toll was invisible to future geologists, for these organisms left no hard parts, no skeletons, and no durable record. Yet the biochemical impact was immense, reshaping ecosystems from the molecular level upward. Where skeletons were absent, chemical signatures in sediments whispered of life extinguished before it could leave durable evidence. Subtle shifts in carbon and sulfur isotopes, coupled with redox-sensitive trace elements, reveal large-scale die-offs of microbial mats and other soft-bodied organisms. Iron and manganese oxidation layers hint at previously abundant life being chemically decomposed before burial. Sedimentary facies preserve only faint echoes of biomass collapse, yet when interpreted in context, they illuminate a planet undergoing mass extinction at a scale rivaling later, more visible events. Even in the absence of fossils, these geochemical footprints allow us to reconstruct the scope of what was lost. Cells succumbed to reactive oxygen species, their delicate membranes rupturing under the stress of an increasingly toxic atmosphere. Lipid peroxidation and oxidative damage interfered with energy production, causing metabolic failure in populations unprepared for the chemical assault. Photosynthetic microbes, once prolific in shallow waters, suffered DNA damage and enzymatic disruption, leaving only fragmented, inactive mats. Anaerobic and facultative organisms faced novel competition and predation pressures as oxygen-permeated environments they had previously dominated. Life at the microscopic scale, often invisible even to contemporaneous observers, was being decimated in ways that would leave no skeletal testimony. Populations of microbial mats and shallow water eukaryotes shrank, their once thriving communities reduced to isolated pockets of survival. These refugia, often restricted to sheltered coastal zones or geothermally warmed areas, provided temporary respite from oxygen toxicity. Yet isolation came at a cost. Limited nutrient inflow, minimal space for expansion, and heightened vulnerability to environmental fluctuations. While some microbes adapted through dormancy or metabolic flexibility, many vanished entirely, leaving fragmented biospheres where once continuous communities had dominated. The gradual, silent collapse reshaped food webs, removing primary producers and altering the foundations of early marine ecosystems. Geographical refuges, warmed by geothermal activity or shielded by ice, became sanctuaries where fragile life clung to existence. Hydrothermal vents, shallow tidal pools, and subglacial meltwater zones offered localized conditions compatible with survival, providing oxygen-depleted microhabitats amid a toxic, frozen world. Microbial mats thrived only in these chemically buffered oases, their continued activity fueling localized nutrient cycles that persisted against the broader trend of biochemical collapse. These patches of life became evolutionary cradles, preserving lineages that would later repopulate the oceans and exploit newly oxygenated habitats. Trace metal distributions and isotopic anomalies in ancient rocks offer indirect glimpses of population collapse across global oceans. Elevated iron oxide deposits, shifts in carbon isotopes, and enriched sulfur signatures indicate periods when oxidative stress and nutrient limitation combined to create widespread mortality. These geochemical markers reveal pulses of extinction occurring over thousands of years, often synchronized with oxygenation events and glacial advances. By studying these indirect traces, scientists can reconstruct the invisible contours of an extinction event that left no bones yet claimed countless lives at the microbial and soft-bodied scale. Ecosystems, once interwoven and dynamic, became simplified, with nutrient cycles slowing under the dual pressures of oxygen and cold. The removal of dominant microbial populations altered elemental cycling, slowing the flow of carbon, nitrogen, and sulfur. Decomposition processes changed, leaving organic matter trapped in sediments rather than recycled through living communities. Oxygen, once a molecular bystander, became both driver and disruptor, reshaping chemical and ecological networks in tandem. Habitats that had once supported dense, interconnected communities now hosted sparse, scattered populations, reflecting the silent severity of the crisis. The extinction unfolded over millennia, a slow chemical attrition invisible to future geologists, yet catastrophic for contemporary organisms. Unlike later mass extinctions marked by impact events or dramatic volcanic episodes, this one progressed quietly, its agents, oxygen, cold, and nutrient limitation, acting at molecular and ecosystemic scales. Life was lost not in bursts of violence, but in incremental, untraceable steps that cumulatively eradicated whole communities. Only by integrating sedimentary chemistry, isotopic analysis, and the study of surviving refugia can we apprehend the true magnitude of this invisible die-off. Surviving lineages displayed remarkable metabolic flexibility, exploiting niches overlooked or inaccessible to their extinct neighbors. Facultative anaerobes, chemoautotrophs, and thermophilic microbes persisted in microenvironments, shielded from the full force of oxygenation. These survivors were the ancestors of later, more complex eukaryotic lineages, laying the groundwork for evolutionary innovation in an increasingly oxygen-rich world. Even in the shadow of widespread mortality, the selective pressure imposed by oxygen forged resilience, setting the stage for future diversification and adaptation across multiple ecosystems. Yet not all life vanished. Some found refuge in the shadows, quietly persisting amid the aftermath of the planet's invisible purge. Subglacial pools, geothermally heated coastal margins, and oxygen-depleted sediments provided havens where organisms endured, maintaining essential biochemical processes and preserving genetic lineages. These surviving pockets, though small and scattered, became the seedbeds for the eventual resurgence of life in oceans now irrevocably altered by chemistry and climate alike. In the silent wake of one of Earth's deadliest yet least visible extinction events, resilience and adaptability emerged as the defining traits of life, hinting at the persistence and ingenuity that would shape the coming ages of our planet's deep history. Sanctuaries in poisoned seas. In the aftermath of widespread mortality, life clung to pockets where chemistry remained familiar. As oxygen permeated oceans and shallow seas, Most organisms confronted environments that exceeded their biochemical tolerances, triggering widespread mortality among soft-bodied and microbial communities. Yet, in isolated zones where oxygen failed to penetrate, conditions remained reminiscent of the planet's ancient, low-oxygen past. These sanctuaries, subglacial meltwater channels, oxygen-depleted sediments, and hydrothermal vent zones harbored pockets of life capable of weathering a planet undergoing profound chemical transformation. Here, microbial mats thickened, early eukaryotes persisted, and complex biochemical cycles continued in miniature, isolated forms that mirrored ecosystems from a time before oxygen's dominance. Sediment layers, shielded from oxygenation, preserved microhabitats rich in nutrients and microbial activity. Fine-grained silt and clay created physical barriers that slowed the diffusion of oxygen into underlying sediments, allowing communities to survive beneath chemically stable strata. These layers concentrated organic matter, sulfides, and iron, sustaining metabolic networks even in the absence of sunlight or surface productivity. Early microbial mats anchored themselves to these nutrient-rich surfaces, forming dense, stratified colonies that maintained localized ecosystems over centuries. In these pockets, life endured through chemical steadiness, exploiting conditions largely unchanged since the planet's pre-oxygenated era, and gradually shaping microgradients that would influence subsequent evolutionary trajectories. Anaerobic microbes exploited chemical gradients, converting sulfides, iron, and other compounds into energy in near-total darkness. Sulfate-reducing bacteria metabolized available sulfur compounds, producing hydrogen sulfide as a byproduct, while methanogens continued limited methane cycles in oxygen-free microenvironments. Fermentation, acetogenesis, and other anaerobic pathways provided critical energy for survival, even as aerobic competitors failed. These chemical transformations not only sustained individual organisms, but also contributed to localized geochemical signatures, preserved as sulfur and carbon isotope anomalies in the rock record. The metabolic ingenuity of these microbes allowed them to thrive where oxygen-intolerant eukaryotes could not, turning chemical adversity into a mechanism for persistence and continuity across millennia. Soft-bodied eukaryotes adapted to these extreme niches, relying on metabolic versatility and behavioral strategies to persist. Early protists and simple multicellular organisms avoided oxidative stress by inhabiting sediment layers, low-light waters, or hydrothermal gradients where oxygen levels remained minimal. Some developed symbiotic relationships with anaerobic bacteria, exchanging metabolites to maintain energy production and reduce oxidative damage. Morphological adaptations, such as reduced surface area and protective extracellular matrices, further limited exposure to oxygen. These lineages represented evolutionary experiments in survival, testing the limits of biochemical flexibility and resilience, while maintaining essential reproductive and metabolic functions under conditions that would have been lethal to other organisms. Hydrothermal vents and subglacial melt zones offered warmth and chemical stability amid a world increasingly hostile to life. Geothermal energy sustained thermal gradients that kept pockets of water liquid despite widespread freezing and oxygenation. Chemically rich fluids provided essential minerals and electron donors, allowing chemosynthetic microbial communities to persist independently of sunlight. Vent-adjacent mats formed intricate ecosystems where anaerobic microbes, archaea, and early eukaryotes interacted, recycling nutrients and maintaining localized food webs. These refuges became evolutionary cradles, preserving lineages that would later seed post-extinction oceans and demonstrate that survival depended not solely on avoiding oxygen, but on exploiting the chemical diversity of these protected habitats. Microbial mats formed dense, layered structures that buffered communities from both oxygen and temperature fluctuations. Stratification within mats created microenvironments with sharply contrasting redox conditions, permitting coexistence of multiple metabolic types, from anaerobic fermenters to facultative sulfur oxidizers. Mats acted as both physical and chemical shields, slowing oxygen penetration while maintaining localized nutrient cycling. Within these dense layers, chemical gradients supported sequential metabolic processes, allowing energy flow and reproduction to continue. These structures preserved genetic and metabolic diversity, ensuring that even within a hostile, frozen, and oxygenated ocean, fragments of life could continue their biochemical work and act as reservoirs of resilience for future evolutionary expansion. Chemical cycling within refuges maintained localized ecosystems, allowing limited but critical reproduction and growth. Carbon, nitrogen, sulfur, and iron compounds were recycled within microgradients, sustaining successive generations of microbes and small eukaryotes. Sulfate and iron transformations fueled energy metabolism, while limited organic matter supported trophic interactions among coexisting species. Though cycles were slow and spatially restricted, they maintained continuity in life's processes and preserved biochemical diversity that had been nearly obliterated elsewhere. These processes illustrate how life can endure under extreme constraints, exploiting narrow niches to survive planetary-scale upheaval while maintaining the foundations of future evolutionary innovation. Isotopic and mineralogical evidence in ancient rocks points to widespread oxygen-depleted zones that served as life's last bastions. Elevated ratios of 34S, 32S, preserved pyrite layers, and iron and manganese mineral distributions indicate sustained anaerobic activity in sediments shielded from oxygen. Trace metals such as molybdenum and uranium remained in reduced states, suggesting oxygen penetration was incomplete and selective. These geochemical fingerprints reveal the locations and characteristics of refuges that allowed lineages to survive a world increasingly hostile at both molecular and ecological scales. Even without visible fossils, the chemistry of sediments speaks to persistence amid chemical adversity, providing a rare window into life's hidden resilience. Isolation, however, came at a cost. Limited space, finite nutrients, and vulnerability to chemical intrusion from expanding oxygenated waters. Populations remained small, fragmented, and genetically constrained, increasing susceptibility to local extinction. Seasonal fluctuations, glacial melt, and diffusion of oxygenated water occasionally overwhelmed these refuges, testing the endurance of surviving communities. Despite these pressures, refuges provided temporary stability, allowing critical metabolic and genetic diversity to persist. Life's survival was precarious, dependent on both environmental fidelity and adaptive flexibility, creating a delicate balance between persistence and eventual exposure to chemical stress. Even as refuges formed, oxygen continued to rewrite biology elsewhere, foreshadowing future challenges for surviving lineages. While isolated communities persisted in chemical time capsules, vast expanses of ocean and shallow habitats were irrevocably transformed, establishing selective pressures favoring aerobic metabolism and oxidation-tolerant organisms. These refuges preserved the echoes of an ancient, pre-oxygen world, yet simultaneously served as launch pads for lineages that would eventually repopulate oceans now irrevocably changed. The persistence of life in these pockets underscores the duality of oxygen as both destroyer and shaper, highlighting that adaptation, innovation, and resilience would become essential traits in the next chapter of Earth's evolving biosphere, the slow birth of cellular defense. Emerging from isolated refuges, some organisms faced a world where oxygen demanded adaptation rather than avoidance. Life that had persisted in chemical pockets now encountered diffuse oxygen invading the seas, exposing cells to reactive molecules that could rapidly destabilize proteins, lipids, and nucleic acids. Survival no longer depended solely on seclusion, but on innovation at the molecular level. Those lineages that managed even minimal protective responses gained a competitive advantage, establishing a subtle but critical evolutionary trajectory. The ocean, once a safe harbor, now imposed a new set of challenges, shaping biochemical ingenuity in ways that would define life's future interactions with oxygen. Within each cell, reactive oxygen species challenged fundamental biochemical machinery. Superoxide radicals, hydrogen peroxide, and hydroxyl species threatened essential metabolic pathways, often in ways invisible to the naked eye. Lipid membranes became sites of oxidative damage, nucleic acids were prone to mutation, and proteins suffered misfolding or functional loss. Organisms without protective strategies were vulnerable to cascading cellular failure. The severity of this intracellular assault underscored the evolutionary imperative: either develop chemical shields or face inevitable collapse in the increasingly oxidized environment. Primitive enzymes such as superoxide, dismutase, and catalase began to appear, neutralizing harmful radicals before irreversible damage occurred. Superoxide dismutase catalyzed the conversion of superoxide into oxygen and hydrogen peroxide, which catalase then broke down into water and oxygen. These enzymatic defenses, emerging in the earliest facultative anaerobes and microeukaryotes, created the first line of molecular protection. Even modest increases in enzyme efficiency significantly extended survival, allowing organisms to tolerate oxygen concentrations that would otherwise have been lethal. These innovations represented a critical evolutionary leap, transforming oxygen from an indiscriminate toxin into a manageable environmental factor. Pigments and small molecule antioxidants complemented enzymatic defense, creating multi-layered protection against oxidative stress. Carotenoids, flavonoids, and glutathione systems scavenged free radicals, preventing cellular components from incurring damage. These molecules not only quenched reactive species, but also absorbed excess energy from sunlight and radiation, offering a dual defense strategy. In some microorganisms, pigment-rich layers formed in extracellular matrices or cell walls, effectively shielding sensitive interior components. The combination of enzymatic and chemical defense created redundancy, increasing the probability of survival in environments where oxygen fluctuated unpredictably and oxidative bursts could strike without warning. Cell membranes underwent compositional shifts, incorporating more resistant lipids to maintain integrity under chemical assault. Saturated fatty acids replaced more vulnerable, unsaturated forms, reducing the likelihood of peroxidation. Membrane-bound proteins evolved to sense oxidative stress and initiate repair processes, while lipid microdomains compartmentalized vulnerable reactions. By fortifying the cell boundary, organisms established both a physical and chemical buffer, allowing internal metabolism to proceed relatively undisturbed despite external oxygen exposure. This subtle restructuring exemplifies how life adapts not by changing location, but by transforming the very architecture of survival at the molecular level. Some lineages developed metabolic compartmentalization, isolating oxygen-sensitive reactions from the surrounding cytoplasm. Organelles, or membrane-bound microenvironments, restricted the exposure of fragile enzymes and pathways to reactive oxygen species, effectively segregating high-risk reactions from the cytosolic milieu. Such compartmentalization allowed cells to continue essential processes such as DNA replication, amino acid synthesis, and energy metabolism without succumbing to oxidative damage. This early spatial organization foreshadowed the more complex organelle systems of later eukaryotes, demonstrating that structural innovation often paralleled chemical adaptation and that life could thrive not just through biochemical tools, but also through the strategic organization of its internal environment. Fossil and isotopic evidence hints at early biochemical adaptations preserved in lipid residues and trace element distributions. Specific ratios of carbon and sulfur isotopes in ancient sediments suggest sustained anaerobic metabolic activity alongside emerging oxygen-tolerant lineages. Lipid biomarkers indicate the presence of pigment molecules and antioxidant systems in microorganisms that survived oxygen exposure. Redox-sensitive metals, including molybdenum and manganese, exhibit patterns consistent with enzymatic activity aimed at neutralizing reactive oxygen species. These geochemical signatures provide a tangible record of life's incremental innovations, demonstrating that even the smallest molecular adjustments could have far-reaching evolutionary consequences. Environmental pressures, including oxygen fluctuations, thermal gradients, and competition, drove rapid refinement of molecular shields. Localized oxygen influx, seasonal circulation, and chemical heterogeneity selected for cells capable of responding flexibly to oxidative stress. Populations that could upregulate antioxidant enzymes, modify membrane composition, or compartmentalize reactions effectively outcompeted less resilient neighbors. The pace of adaptation, while slow on a human scale, was rapid in evolutionary terms, as incremental improvements conferred immediate survival benefits. Life was learning not merely to endure oxygen, but to integrate it into the chemical rhythms of existence. Despite these innovations, early defenses were imperfect and survival remained precarious. Oxidative bursts still caused mortality. Metabolic repair mechanisms were energetically costly, and environmental variability constantly tested cellular resilience. Populations persisted in fragmented refuges or in niches with limited oxygen penetration, balancing the need for exposure to nutrients against the threat of oxidative damage. Yet these incremental adaptations laid the foundation for more sophisticated control over oxygen metabolism, illustrating a key principle. Survival did not require perfection, only sufficient adaptation to navigate an increasingly hostile environment. The slow accumulation of defensive strategies set the stage for the next evolutionary leap. These first cellular shields marked the beginning of life's mastery over oxygen, a prelude to mechanisms that would eventually harness it as a tool rather than a threat. Early antioxidant systems, pigments, and structural modifications transformed oxygen from a lethal poison into a factor that could be tolerated, giving rise to ecosystems capable of gradually expanding into previously uninhabitable waters. This molecular evolution established the principle that defense was only the initial step. True control over oxygen, with the potential for energy-intensive aerobic metabolism and broader ecological dominance, lay ahead. Life had begun to convert vulnerability into opportunity, signaling the slow but inexorable ascent toward a biosphere shaped not just by survival, but by the mastery of one of Earth's most reactive elements. Energy unleashed, danger included. Life that had merely survived oxygen now began to exploit it as a potent source of energy, transforming survival into opportunity. Early facultative aerobes, including primitive cyanobacteria and emerging proto-eukaryotic lineages, harnessed the chemical potential of oxygen to fuel higher metabolic rates. These cells could reproduce faster, move more effectively, and colonize oxygenated microenvironments, giving them a competitive edge over strictly anaerobic neighbors. In patchy coastal pools and shallow seas, microbial mats thickened and expanded, forming dense communities where energy-intensive processes such as motility and nutrient acquisition became possible. Oxygen had shifted from a chemical threat into a driver of ecological innovation, shaping the very architecture of early life. Harnessing oxygen enabled far greater energy extraction per molecule than fermentation, opening new pathways for growth and complexity. Whereas anaerobic fermentation yielded a mere 2 ATP molecules per glucose, aerobic respiration could provide up to 36, empowering cells to maintain larger sizes, more elaborate intracellular structures, and active movement. Colonies of motile aerobes could sweep across nutrient-rich patches, outcompeting slow-growing anaerobes and reshaping microbial community composition. Even small energy advantages translated into significant ecological dominance, allowing aerobic lineages to colonize previously marginal zones and establish early ecological hierarchies. The chemical revolution of oxygen became inseparable from the emerging story of life's expansion. Electron transport chains allowed cells to release energy incrementally, balancing opportunity with risk. Cytochromes, iron-sulfur clusters, and quinones coordinated electron flow toward oxygen while minimizing the formation of destructive radicals. In habitats ranging from shallow tidal pools to oxygen-enriched sediments, these molecular systems gave aerobic organisms the ability to exploit energy-rich niches inaccessible to their anaerobic competitors. Even small proto-eukaryotes with compartmentalized organelles benefited, using electron transport not only for energy, but also for early environmental sensing, motility, and adaptive behavior. The molecular choreography of electron flow, once invisible, now had profound ecological consequences. The proton gradients generated by these chains powered ATP synthase, producing energy to sustain growth, movement, and adaptation. Microbial colonies expanded faster. Motile cells navigated toward nutrients and away from harmful conditions, and early proto-eukaryotes could maintain internal homeostasis across larger cell volumes. This energetic advantage allowed certain lineages to dominate oxygenated patches, creating hotspots of activity that altered nutrient flux, redox gradients, and the spatial distribution of life. Oxygen was not just a fuel, it became a selective agent, reshaping community structure and giving rise to the first localized ecological competition influenced by chemical energy availability. Facultative and obligate aerobes exploited these advantages to colonize new habitats and outcompete their anaerobic neighbors. Early proto-eukaryotic organisms, possibly akin to modern amoeboid forms, moved toward oxygen-rich microenvironments, where higher energy yields allowed faster replication and more complex behaviors. Microbial mats thickened in oxygenated zones, forming layered communities where resource cycling became more efficient and competitive interactions intensified. Even at the microscopic scale, selective pressures were amplified, favoring lineages that could both tolerate oxygen and exploit it to grow, divide, and move faster than surrounding populations. Life had begun not only to survive, but to strategically dominate. Yet energy came with cost. Reactive oxygen species threatened the very structures that allowed aerobic metabolism. Superoxide, peroxide, and hydroxyl radicals damaged DNA, proteins, and lipids, creating a constant selective pressure. Organisms with superior antioxidant systems, repair enzymes, or behavioral strategies to avoid high oxygen concentrations were more likely to thrive. Early motile colonies, for instance, could migrate to optimal oxygen zones while minimizing radical damage. Evolution rewarded ingenuity, balancing the enormous benefits of energy exploitation with the persistent dangers of reactive chemistry. Antioxidant systems and molecular compartmentalization evolved in response, allowing life to safely harness oxygen. Superoxide dismutase, catalase, and peroxidases neutralize radicals, while membrane-bound compartments shielded sensitive reactions. Even pigments and small molecule scavengers played a role in defending delicate macromolecules. These defenses enabled aerobic organisms to grow, divide, and move without succumbing to oxidative stress, giving rise to the first energy elites in microbial ecosystems. Oxygen became not merely a threat to be endured, but a tool to be wielded, fostering ecological specialization and resilience. Energetically privileged organisms transformed ecological landscapes, establishing patterns of dominance and exclusion. Motile aerobes could explore oxygenated sediments, trace nutrient gradients, and colonize microhabitats inaccessible to slow-growing anaerobes. Microbial mats became patchy but dynamic, structured by competition between oxygen-tolerant and oxygen-sensitive lineages. Early predator-prey-like interactions, such as the consumption of smaller cells or scavenging within mats, were now possible because of the surplus energy available to motile aerobes. These emerging interactions hinted at the complex ecological webs that would characterize later oxygenated oceans. The distribution of life itself began to reflect oxygen availability, with winners in enriched zones and losers confined to refuges. Populations capable of rapid aerobic metabolism could expand, reproduce, and explore more widely, while anaerobic lineages persisted only in oxygen-depleted pockets. Environmental heterogeneity, combined with chemical stress, created a patchwork of thriving and struggling communities. This uneven distribution amplified selection pressures, accelerating evolutionary innovation among survivors and magnifying disparities between regions. Oxygen was now a central architect of early ecological organization. By converting a once lethal gas into a source of power, life fundamentally reshaped ecological competition. Energy-intensive metabolism allowed certain lineages to dominate local habitats, innovate behavioral strategies, and exploit resources with unprecedented efficiency. Communities were no longer shaped solely by scarcity or survival, but by the capacity to harness oxygen, giving rise to the first biologically driven energy hierarchies. Cells had transformed vulnerability into advantage, chemical threat into opportunity, and this imbalance began reshaping competition itself, laying the groundwork for the next era of ecological complexity: winners, losers, and an uneven Earth. Across oxygen gradients, life began to reveal stark disparities. In shallow seas and coastal margins, oxygen-rich pockets allowed certain lineages to thrive, while nearby anaerobic refuges became islands of struggle. Motile proto-eukaryotes and facultative aerobes expanded into these enriched zones, their elevated metabolism providing the energy to reproduce faster, explore microenvironments, and monopolize resources. In contrast, obligate anaerobes faced lethal conditions where oxygen accumulated, forcing retreat into sediments, tidal pools, and oxygen-depleted basins. The planet's first chemically driven ecological mosaic had emerged, with survival dictated as much by location as by evolutionary ingenuity. Some proto-eukaryotes expanded rapidly, propelled by newfound energetic advantages. Small, amoeboid-like organisms could actively move through microbial mats, positioning themselves in optimal oxygen niches and grazing on slower anaerobic cells. Cyanobacterial mats thickened in oxygenated shallow waters, creating microecosystems where productivity soared, nutrient cycles intensified, and colony structures became increasingly complex. Energy efficiency allowed aerobic populations to maintain larger sizes and more sustained growth, gradually dominating patches of the seafloor and forming the earliest competitive hierarchies based on chemical resource access. In contrast, anaerobic microbes persisted only in isolated refuges where oxygen remained scarce. Deep sediments, subtidal basins, and oxygen-poor channels became the final bastions for lineages that had once dominated the seas. These environments, while protective against oxidative stress, imposed severe limitations on growth, mobility, and reproduction. Populations shrank, genetic diversity declined, and previously abundant species vanished from large swaths of the planet. The uneven distribution of life highlighted that adaptation to oxygen was no longer optional. It was the determinant of survival. Layered microbial mats in shallow waters became arenas of competition, cooperation, and selective pressure. Cells capable of tolerating oxidative stress gained first access to light, nutrients, and space, while sensitive species were excluded or forced to retreat. Within these mats, gradients of oxygen, sulfide, and nutrients created intricate microhabitats, fostering localized interactions that shaped community composition. Dense mats, sometimes spanning meters, became incubators of innovation, where oxygen-driven metabolism powered faster growth, motility, and even primitive cooperative behaviors among microbial colonies. Motile aerobes exploited chemical microenvironments, outcompeting slower or sedentary lineages. Flagellated proto-eukaryotes and filamentous cyanobacteria maneuvered toward oxygen-rich regions, leaving anaerobic competitors confined to shadows and crevices. Rapid reproduction in these enriched zones reinforced dominance, and colony expansion created positive feedback loops. More aerobic cells produced more oxygen locally, further disfavoring anaerobic neighbors. Life was reorganizing itself not through catastrophe alone, but through the subtle interplay of chemical advantage, energy use, and mobility. Localized extinction events left behind chemical footprints in sediments, hinting at widespread ecological turnover. Iron sulfide layers and redox-sensitive minerals recorded where oxygen had reached lethal concentrations, marking the former territories of anaerobic communities. Stable isotope ratios, preserved in sedimentary rocks, reveal the uneven survival of lineages across different regions, confirming that oxygen acted as both an energy source and an ecological filter. While life persisted globally, these chemical traces bear witness to the first large-scale, patchy reshaping of ecosystems by a single element. Colonization patterns reflected the uneven distribution of oxygen, creating dynamic mosaics of life. Habitats varied in oxygen saturation from nearly anoxic to modestly enriched, and the species composition mirrored these gradients. Proto-eukaryotes expanded outward from high-oxygen microhabitats, forming corridors of occupancy that linked scattered refuges. Meanwhile, anaerobic communities retreated or fragmented, preserving diversity only in isolated patches. This patchwork created evolutionary pressures favoring mobility, energy efficiency, and tolerance mechanisms, establishing the earliest patterns of differential survival that would echo through deep time. Energy-intensive metabolism amplified disparities, allowing some lineages to monopolize resources and shape community structure. Aerobic organisms could exploit nutrients more rapidly, colonize substrate surfaces, and even alter local chemistry, producing microenvironments inhospitable to anaerobic neighbors. Resource partitioning intensified, spatial hierarchies emerged, and the first ecological winners and losers became apparent. Oxygen had transformed from a molecular threat into a landscape-shaping force, establishing a precedent for selective pressures that would continue as life evolved complexity and new ecological interactions. Even within surviving populations, adaptations emerged to balance energy extraction with oxidative stress mitigation. Early antioxidant systems, membrane-bound compartments, and protective pigments allowed aerobic cells to endure variable oxygen levels while maintaining high-energy metabolism. Communities became structured around these adaptations. Tolerant lineages clustered in enriched patches, while sensitive species lingered at the periphery or in refuges. The combination of energy advantage and chemical resilience defined success, creating the first ecosystems where metabolism dictated both survival and territorial influence. Amid these emerging oxygen mosaics, larger microbial colonies began exploiting spatial separation for survival. Filamentous organisms extended networks into oxygenated and anoxic zones simultaneously, balancing energy gain with oxidative risk. Biofilm architectures became increasingly sophisticated, incorporating layers of specialized cells, some photosynthetic, others protective or detoxifying. These structures not only maximized resource use, but also created miniature oxygen refuges within otherwise hostile surroundings, exemplifying the earliest instances of spatially organized survival strategies. At the same time, regional variability in oxygen exposure produced divergent evolutionary pathways. Lineages in oxygen-rich zones evolved traits favoring motility, nutrient uptake, efficiency, and intercellular cooperation, while those in anoxic refuges conserved ancestral metabolic pathways, slow growth, and dormancy capabilities. This divergence established the blueprint for long-term ecological differentiation. Some populations would eventually give rise to energy-intensive aerobic eukaryotes, while others would remain as slow-growing anaerobic forms, isolated yet persistent. The interplay of oxygen, energy, and adaptation shaped both immediate survival and the evolutionary potential of these lineages. Differing in occupancy and productivity, regions gradually diverged, leaving the stage for future evolutionary trajectories. Oxygen-rich zones nurtured innovation, motility, and accelerated reproduction, while oxygen-poor refuges preserved ancestral lineages under protective isolation. The uneven biosphere was no longer a uniform canvas, but a mosaic of opportunity and constraint, where survival depended on chemical mastery, energy utilization, and the capacity to exploit localized advantages. Life had entered an era defined not by uniformity, but by disparity, and the first true patterns of winners and losers were etched across Earth's oxygen-altered landscapes, a planet out of balance. Regions that had flourished in oxygen-rich environments now faced chemical instability as the planet struggled to reconcile the rapid transformations imposed on its biosphere. Shallow seas, long considered stable cradles for microbial life, oscillated between oxidizing and reducing conditions, each shift subtly altering which organisms could persist. Cyanobacterial mats, once prolific architects of oxygen production, found themselves both creators and victims of the very gas they emitted, their metabolic bursts intensifying chemical feedback in local waters. Meanwhile, early motile proto-eukaryotes exploited ephemeral oxygen-rich niches, their locomotion and feeding strategies, allowing them to outcompete slower, anaerobic counterparts in patches that fluctuated in redox balance. Life was no longer shaped solely by uniform survival pressures. Instead, success depended on navigating a fractured, unpredictable chemical landscape. Shallow tidal flats became theatres of both opportunity and collapse. Evaporation concentrated salts and minerals, amplifying oxidative stress in organisms that had not yet evolved protective mechanisms. Microbial mats flourished in certain areas, forming dense layers that temporarily buffered communities from sudden spikes in oxygen. Yet these mats could collapse overnight under chemical assault. In response, anaerobic microbes retreated to subtidal basins and sediment pockets where oxygen intrusion was minimal, carving out refuges that preserved ancient metabolic pathways. These dynamic spatial patterns revealed the uneven nature of survival, where life clung to chemical niches as much as to physical habitats, and where patchy oxygen levels dictated local biodiversity. Seasonal variations further intensified this chemical imbalance. Ocean circulation, tidal fluxes, and temperature gradients cause rapid swings in oxygen concentration, creating ephemeral windows in which aerobic organisms could expand rapidly, only to face sudden die-offs when oxygen levels spiked beyond tolerance. In some regions, iron and sulfur cycles intersected, producing reactive compounds that added an invisible layer of threat to surviving lineages. Filamentous cyanobacteria and early eukaryotic cells experienced both boon and hazard as nutrient availability coupled with redox conditions to produce boom and bust population cycles. Even organisms that had evolved rudimentary antioxidant systems were tested by the speed and scale of these environmental oscillations, revealing the limits of early cellular defenses. Subsurface sediments emerged as critical refuges, harboring organisms protected from surface volatility. These oxygen-depleted pockets allowed anaerobic bacteria to persist, maintaining metabolic strategies that had dominated the planet before oxygen's rise. Fossil evidence from iron-rich stromatolites suggests that microbial communities in these refuges remained remarkably stable compared to their oxygen-exposed counterparts. Yet, even here, chemical interactions continued quietly. Diffusing oxygen from overlying waters occasionally penetrated, creating microgradients that favored organisms capable of tolerating intermittent exposure. Survival demanded both biochemical flexibility and strategic occupation of the most stable microhabitats available. Cyanobacterial activity became a double-edged sword in these fluctuating systems. Their photosynthetic oxygen production catalyzed chemical reactions in the surrounding waters, oxidizing dissolved iron and sulfides, and triggering the precipitation of rust-colored iron minerals. These reactions, while sequestering potentially harmful compounds, also altered nutrient availability and pH, further shaping which organisms could thrive. Motile proto-eukaryotes exploited transient oxygenated zones, feeding on microbial prey or scavenging detritus, while static anaerobes were forced into marginal refuges. The interplay between metabolic output and environmental consequence revealed how biological innovation itself could amplify instability across the landscape. Across broader scales, These patchwork conditions created stark contrasts in reproductive success. Aerobic microbes expanded rapidly in regions where oxygen and nutrient fluxes aligned, reproducing multiple generations in the time anaerobic populations produced one. This disparity amplified differences in biomass and resource consumption, setting the stage for cascading ecological effects. Zones of die-off released nutrients and detritus that temporarily fueled survivors, creating cycles of boom and collapse that shaped the very structure of early ecosystems. Life had become a dynamic experiment in adaptation, where chemical gradients, rather than geography, dictated ecological opportunity. Feedback loops between oxygen, methane, and iron added another layer of unpredictability. Oxygen reacted with methane, reducing greenhouse warming and initiating localized cooling events, which in turn affected solubility, circulation, and redox chemistry in shallow seas. Iron availability influenced both microbial metabolism and the precipitation of minerals that could temporarily buffer oxygen fluxes. These interconnected cycles produced a planet-wide mosaic of chemical stability and instability, where even small variations could tip local habitats from prosperity to near collapse. Survival was contingent not merely on tolerance, but on an organism's ability to respond to cascading environmental consequences. Competition intensified as organisms adapted to specific niches. Aerobic microbes exploited high oxygen patches with energy-intensive metabolisms, enhancing motility, growth rate, and resource acquisition. Anaerobic survivors, meanwhile, specialized in low oxygen refuges, metabolizing sulfur or other reduced compounds unavailable to oxygen-tolerant organisms. This uneven distribution created the first true energy hierarchies in microbial ecosystems, where metabolic efficiency became a determinant of ecological dominance. Predation, scavenging, and microbial grazing intensified within these energy-rich pockets, further differentiating winners from losers in a biosphere governed by chemical gradients. Temporal instability compounded spatial heterogeneity. Fluctuations in oxygen and nutrient availability led to episodic population crashes and recoveries, forcing rapid evolutionary experiments. Some proto-eukaryotes evolved membrane-bound organelles capable of compartmentalizing oxidative stress, while certain bacterial lineages reinforced enzymatic defenses against reactive oxygen species. Communities capable of rapid adaptation dominated transiently, while less flexible organisms were consigned to shrinking refuges or extinction. Over successive generations, these pressures sculpted increasingly complex interactions, laying the groundwork for ecosystems defined by chemical stratification and energetic hierarchies. Over millennia, these compounding factors transformed Earth into a planet of contrasts. Chemical instability, uneven oxygen distribution, and feedbacks between metabolism and mineral precipitation prevented the biosphere from achieving equilibrium. Early aerobic lineages became widespread yet remained vulnerable to sudden shifts, while anaerobic refuges preserved relic strategies in pockets of stability. Life was neither uniformly flourishing nor universally collapsing. Instead, it occupied a mosaic of opportunity and constraint, where success required not only adaptation but constant vigilance. As landscapes diverged and ecosystems oscillated between abundance and scarcity, time amplified these differences, quietly forging a world in which chemistry, climate, and biology were forever entangled, and a radically altered biosphere was emerging from the imbalance. Life under a changed sky. Following thousands of years of chemical upheaval, life confronted an atmosphere no longer optional but omnipresent, where oxygen had shifted from occasional threat to permanent environmental factor. The gas that had once silently wrought havoc on unprepared cells now dictated the very strategies of survival, influencing energy acquisition, mobility, and reproductive timing. Organisms that had survived in scattered refuges, shielded by sediments, suboxic waters, or geochemical barriers, found themselves under persistent pressure to integrate oxygen into their metabolism. Those unable to tolerate it vanished, while others began to experiment biochemically, transforming adversity into opportunity. Even the most subtle chemical gradients shaped the evolutionary landscape, setting the stage for innovations that would reverberate for eons. Microbial pioneers refined their metabolism in response, developing enzymatic shields capable of neutralizing reactive oxygen species while still extracting energy efficiently from organic substrates. Superoxide dismutase, catalase, and peroxidase systems emerged as crucial molecular innovations, reducing oxidative damage within cells and enabling longer lifespans and higher reproductive output. These adaptations allowed aerobic microbes to colonize previously uninhabitable regions of shallow seas and coastal margins, where oxygen levels fluctuated but never vanished entirely. As these organisms expanded their ecological footprint, they introduced both competition and opportunity, fostering the first biological hierarchies influenced directly by oxygen availability, rather than merely by nutrient abundance. Sediment-dwelling anaerobes, the relics of a pre-oxygen world, persisted in protective niches, yet even these pockets were not entirely insulated. Intermittent exposure to rising oxygen levels demanded dynamic survival strategies. Some formed dormant cysts capable of enduring oxidative stress, while others localized to microenvironments rich in sulfide or other reducing agents. Their persistence demonstrates the duality of this period. Oxygen acted simultaneously as destroyer and selective sculptor, eliminating some lineages while creating ecological space for the adaptive few. Fossil evidence from stromatolite mats suggests that these mixed communities were surprisingly diverse, with aerobic and anaerobic organisms coexisting in chemical mosaics across marine landscapes. Motile proto-eukaryotes began exploiting oxygenated microenvironments in remarkable ways, balancing energy efficiency with oxidative risk. Early flagellates and amoeboid forms migrated between oxygen-poor refuges and oxygen-rich zones to access resources and evade competition, effectively navigating a patchwork of opportunity and danger. Their movement allowed rapid colonization of nutrient-rich habitats, spreading oxygen-tolerant lineages more widely than stationary microbes could achieve. Behavioral flexibility complemented molecular defenses, demonstrating that survival was no longer solely a matter of biochemistry, but increasingly a matter of strategy, timing, and spatial awareness within a chemically divided world. Photosynthetic oxygen production intensified feedback loops in local ecosystems, reinforcing selective pressures on both producers and consumers. Cyanobacterial mats, increasingly prevalent in shallow waters, generated bursts of oxygen that temporarily exceeded ambient concentrations, creating zones of lethal toxicity for unadapted microbes. Yet this very toxicity acted as an evolutionary filter, favoring lineages capable of tolerating or exploiting oxygen, accelerating the spread of aerobic metabolism. By contributing both resource and hazard, these early oxygen producers reshaped the biological stage, making the planet's chemical landscape an active participant in the evolutionary drama. Energy-intensive aerobic metabolism conferred a decisive advantage to those lineages capable of tolerating oxygen, Cells that could exploit this pathway achieved higher energy yields per substrate molecule, enabling faster growth, more frequent division, and greater motility. This metabolic edge translated directly into ecological dominance. Aerobic microbes outcompeted less adaptable organisms for space, nutrients, and access to sunlight, effectively rewiring community structures. Even minimal differences in oxygen tolerance became amplified over generations, gradually establishing a biosphere organized around chemical capacity rather than sheer abundance or predation alone. Competition sharpened within these oxygen-influenced environments. Early aerobic lineages dominated local habitats, not merely through energy efficiency, but through innovation in resource utilization, including the ability to metabolize previously untapped organic compounds or colonize exposed surfaces. Predatory protists, feeding on smaller bacteria, took advantage of oxygen-rich zones to enhance their own metabolic throughput, establishing feedback loops of selective pressure and population expansion. In turn, anaerobic lineages retreated further into marginal niches or sedimentary refuges, creating stark contrasts between thriving and struggling communities. The unevenness of survival became a defining feature, signaling that chemical capacity now dictated ecological hierarchy. Genetic innovation spread through these populations with remarkable speed, embedding oxygen tolerance into the very fabric of cellular life. Horizontal gene transfer, mutation of protective enzymes, and the emergence of compartmentalized organelles in proto-eukaryotes all contributed to more resilient metabolisms. By integrating oxygen into core cellular processes, life effectively transformed a once lethal gas into a tool for growth, movement, and reproduction. This molecular and genetic revolution laid the foundation for future eukaryotic complexity, establishing the biochemical infrastructure for multicellularity, predation, and eventually terrestrial colonization. Ecosystems stabilized into mosaics of oxygen-tolerant dominance and protected refuges, where chemical gradients created microhabitats with divergent selective pressures. Some shallow marine regions flourished with aerobic productivity, supporting proto-food webs and fostering innovation in motility and predation, while deeper sediments and anoxic pools preserved ancestral anaerobes. The coexistence of these divergent communities created a layered biosphere, where evolutionary pressures operated on multiple levels, simultaneously: molecular, behavioral, and ecological. Life was no longer merely surviving. It was organizing, strategizing, and exploiting the uneven world that oxygen had wrought. By this point, the planet had entered a new equilibrium, one defined not by the absence of challenge, but by adaptation to a chemically transformed environment. Oxygen-tolerant lineages expanded across oceans and shallow seas, while anaerobic relics lingered in constrained refuges, producing a biosphere marked by contrast, resilience, and diversity. Energy utilization, defensive biochemistry, and behavioral flexibility had become intertwined, shaping survival into a dynamic interplay of advantage and constraint. The chemical, ecological, and evolutionary landscapes were permanently coupled, ensuring that no organism could ignore the presence of oxygen without dire consequences. Ultimately, survival had become inseparable from adaptation to oxygen, and the lessons of this era would echo across deep time. Organisms had transformed vulnerability into advantage, exploiting the reactive power of oxygen for energy while mitigating its destructive potential through molecular, behavioral, and ecological strategies. The sky had finished its transformation, but life was only beginning to understand what it had endured, and the biosphere it inherited would never again be the simple, anaerobic world it had once been. The story of oxygen, once a silent killer, has now become the story of life itself, energetic, adaptable, and poised to explore evolutionary horizons previously unimaginable. The air that once suffocated became the crucible in which life forged resilience, innovation, and the first whispers of complexity. In the glow of a transformed sky, survival was no longer chance, it was adaptation, ingenuity, and courage at a microscopic scale. Every molecule of oxygen carried both threat and promise, reminding us that life's greatest challenges often sculpt its most profound triumphs. As the planet exhaled its ancient, reactive breath, the first true architects of a new biosphere emerged. If this journey through deep time captivated you, feel free to like, subscribe, or share your thoughts below.